Fourth part of this lecture, we will be discussing about the optical bioimaging and a brief introduction to MRI. So, optical bioimaging is of course the first bioimaging technique that was developed. So, it involves, it has a long history, so it involves microscope, of course. So, they are the first microscope and the first bioimaging techniques that were developed. So, the microscopes were first developed during the Renaissance, that's around the 14th to the 16th century, by using convex glass to magnify images, the objects and images. So in here, you have a brief or a very simple schematic of how how a simple magnifying lens can actually produce an image, a magnified visual image of your subject. So of course, there's something to do about the bending of light. So the magnifying lens is capable of bending the light such that you will form a larger visual image that can be perceived by your eyes. Now, the most common, of course, for optical microscopy is, of course, a compound microscope, which just uses light. So we usually use that in biomedical engineering part in doing, actually in medicine, it's usually used for histological samples. So you stain tissues, usually biopsies, and then you view it under the microscope. Now, on a more other imaging techniques or optical imaging techniques that are a bit more complicated than your compound microscope is your fluorescence and confocal imaging. So we have fluorescence microscopy. It uses fluoropores. So fluoropores are molecules that emit light at a particular wavelength after excitation. So if you expose them to a certain shorter wavelength of light, for example, blue light or UV, it will fluoresce. It will emit a different light of indifferent intensity or wavelength, so different color. So it will fluoresce, and that fluorescence can be captured by your microscope. So in this fluorescence, if you use special microscope, the same way we do CT scan and of course 3D ultrasound, if you have multiple detectors at different angles, you can form three-dimensional images. So confocal microscopes are more of a level up version of your light microscope. So they allow imaging of multiple levels through a specimen, leading you to a three-dimensional image. So here is an example of your microscope. So this is a fluorescence microscope. So fluorescence microscope, where is the one on the left, is the fluorescence microscope. So in here, your objective or your specimen contains the fluorophore. So it has the fluoropores. So it's either the cells, the cutting or the tissues or the smear on that slide is stained with a fluoropore fluorescent dye that sticks to your tissues or to the cells on the tissues. And then you have an external light source that you will project a certain a light of a certain wavelength to your specimen. So of course, it has a fluoropore. The fluoropore will then fluoresce, so it will emit light. That emitted light will be detected by your detector. So the setup here is that the light source is perpendicular to your detector. So you have 90 degrees. So that is so that your light source, the external light source, will not interfere with the light coming from your specimen. So that's for fluorescence microscope. And then this is confocal microscopy. So it also uses, this is a fluorescence microscope, but it uses multiple emissions, or rather it scatters the emissions a bit so that your specimen, you can produce a more detailed view of your image. So you form your dichroic mirror. So here is an image of a mouse that contains fluorescent dye. So it was injected with two colors of dye. So one good thing about fluorescence microscopy is that you can inject multiple dyes at the same time, unlike in nuclear medicine, whereas usually it's only one, so the output is based on the emission of a single entity. But in here, you can have two different signals at the same time. So in here, you have your GFP fluorescence, and another one is luciferase fluorescence. So both of them will adhere to a certain tumor. So two different types of tumor. So you can see here the different tumors. So here is an experimental surgical technique they are trying to develop. Actually, it's not yet in use for humans, but it was proposed before. They wanted to use fluorescence microscopy in order to identify the tumors, because the fluorescence dye will stick to the tumor. So the surgeons will now know where the tumor is. And unlike the normal surgery procedures where you excise the tumor, sometimes a bit is left. That's usually the common problem. Now, in here, since it fluoresces, so it's easy to detect if there's any small amount of cells that are left over from the tumor. So you have that. So this is a type of new type of surgical procedure still in the experimental stage. Now, this is a confocal image of your neuron as a nerve cell. So, in confocal microscopy and fluorescence microscopy, you are looking at small or microscopic images. Unlike in the MRI and the CT scan, we are looking at organ levels. So, it's large images, or at least the real-time images. Here, we are looking at very, very small cellular level. So, here is how the confocal microscopy works. So you create a series of images using different focal planes, and then you build them up to create a three-dimensional image. So here is a hippocampal neuron. So you build it up and you form the image on the right, which is your three-dimensional image. Now, for magnetic resonance imaging, I'll just give you a brief intro here, but I will attach additional videos because MRI is it's a bit more there's some more to say about how MRI works. Now, here is a magnetic resonance images. Actually, if you look at them, it's quite similar to CT scan images. The only difference is that in CT scan images, the one we are looking at here is more of a density. So in CT scan, since it's based on X-ray images, you can see the bone clearly. However, in MRI, it's not that clear, or rather, MRI makes use of water molecules. So in MRI, the signal is coming from water molecules, the movement of the hydrogens in the water. Since most of us, 75% of water, human body is made of 75% water. It's a lot of water. So the position of the density of the water molecules in each organ, each body part, is detected by MRI, and that is what makes these images. That's how MRI generates image. It just detects where water is. So in here, you can see, for example, the gray and the white matter in the brain. Here is the 3D image of your brain. So you can clearly see here the brain tissue. So MRI is a good tool to use if you want to image soft tissues, because it produces a more clear pictures with greater resolution. You can see greater details, because since it's water, not all tissues contain the same level of water. So the differences is very apparent in MRI. Unlike in CT scan, which uses X-rays, we are looking at densities, tissue density. So if there are two tissues or organs with similar densities, so it's a bit difficult to distinguish. However, in MRI, it's very difficult to see bone structures, because bones are dense, they are relatively dry, they do not, if you notice, you have a black outline in your skull. That's basically the outline for the skull, so it's not that clear to see because it's dry technically compared to other parts of your body. So the CT scan is usually used for if you want to see the denser portions of the tissues. And then you have MRI, which is for the softer tissues, and it's also more favored because MRI does not produces ionizing radiations. In fact, it just detects the magnetic gyrations of the molecules that's already inside. So it also has another leg up to versus nuclear medicine, because you don't need, in fact, to use any other radioactive probes. Now, the downside here is that since it uses a large amount of magnets, so the mechanism is a bit claustrophobic, of course, and you have your you cannot subject a person that has iron inside, or rather has steel or metal trappings, for example, metallic legs, or you have metallic implants or braces. You cannot subject those people in MRI. At least you need to remove those metallic implants first, because it will jeopardize the machine, because it's very, very sensitive to or rather MRI produces strong magnetic fields, which your metallic implant is very susceptible to. So that's one of the downsides. And then the machine itself is very noisy. So if you ask those, they are very, very noisy. So further info on how exactly MRI detects images will be added into your Blackboard, additional videos outside of this one. Now, here is a type of digital imaging. So basically, this is bioimaging in the future. So what can we expect about bioimaging? So one of the main focus in bioimaging is to increase the resolution, to enhance the resolution of images, because of course the more details the doctor has, the physicians have, the more information they have, and the more accurate their diagnosis will be. So in here is one of the methods. This is actually a satellite view. So one of the way to increase resolution is to use quantized to break down the image into pixels, and then you form solid blocks, and then for each block you can actually refine the image more. So another one here is basically superposition of PET scan and MRI. So this is two types of bioimaging techniques using in tandem with each other. So this is functional MRI. So you have on the right is the structural image, because when you do structural MRI, or even CT scan, you just see the structure, but you do not know how it functions. Is it active right now? Is it inactive right now? We do not know. Now, if we just do PET scan, for instance, PET scan will only give signals on the active sites or the active body parts. We do not know the structure. We just know, there is a part here that is active, but we don't know that. Of course, the resolution is very bad. Now, if we use the information from both, that is, if we can superimpose the PET scan with the CT scan or the structural MRI image, you overlay it, and then we have now the image on the right, letter B, which shows us your brain, the inside of your brain, and in which specific region of the brain is active at a certain moment. That's basically functional MRI. And in psychology, they are using that technique in order to know which part of the brain is being stimulated, or rather is being activated when they give different stimulus. For example, different sense of smell, a large sound, a mellow sound, and then it will light up. They call it the light up different parts of the brain, because the signals will be detected from those specific regions. So that's it for bioimaging techniques. So see you in the next lecture.